Hi, I'm Alexis Gerritsen. I'm a graduate student in the Department of Biology at George Mason University, and today I'm going to be talking to you about extracting phenology and life history data from digitized specimens. The overall thesis of my talk is that applying citizen science protocols to our digital museum specimens can enable further data integration, both for the citizen science data products and for digital museum specimen data products, while enabling broad-scale trend discovery over wide spatiotemporal gradients, particularly in the environmental sciences. So why citizen science protocols? Citizen science data is a growing resource for researchers everywhere, and it's often extremely spatially extensive, particularly in the United States, but it's often limited by the age of the project. So most citizen science projects, with some notable exceptions, are from 2000s or later, which means that there isn't much ability to sort of turn back time and see how trends have changed over a much longer time period. Similarly, when you derive environmental data or phenology data from herbarium products, they're often difficult to validate, and it's difficult to disentangle collection biases and problems with storage. So age of a specimen can sometimes look like things like autumn coloration or other phenological stages. So it can be difficult to validate these herbarium-derived products without having some real-world ongoing correlate. So having this parity allows for that kind of analysis to occur. The third major one is that citizen science protocols often rely only on visual information. So many citizen science projects are observational in nature, meaning that the citizen scientists are expected to go out and take photos or observe or look directly at the things occurring around them. So it's less common that citizen science protocols rely on things like direct collection of specimens or genetic data or other types of collection of specimens. So being able to rely on this visual information means that we can apply it to some of these digitized image stores resulting from the large-scale digitization of museum and herbarium specimens. So I'm going to walk you through two case studies of what this might look like in practice. The first is a phenology question, and in this case we used the National Phenology Network Nature's Notebook protocols. So the Nature's Notebook is a pretty well-known citizen science project where individuals go out, look at the trees, insects in their backyard, and see over the course of the year when are different phenophases or life stages occurring. So for example, if there's a maple tree in your backyard, you might look and see, are there leaves? Are there colored leaves? Are there evidence of fruit or seeds? And they have explicitly designed protocols for a couple thousand plants and animal species with different delineations about what to look for, species specific, to determine whether a phenophase is occurring or not occurring. In addition to presence-absence phenophases, they also have some percentage, what percent of the tree is currently red or in its autumn coloration, and what percent of the tree currently has leaves on it, that kind of thing. So there's both binary and continuous variables. So we applied these protocols directly to herbarium specimens. Only about 8 of them were directly applicable, so we couldn't do things like what percent of the tree is in its autumn coloration, because we often only had 1 or 2 leaves per sheet. But we're able to get quite a few of these phenophases, the presence or absence of leaves on the page, whether there are leaf buds, how many leaf buds, are there breaking leaf buds, and this is something that's collected by NPN, but we actually use some modified protocols from Project Budburst, which is another very large-scale citizen science project collecting observational data. One of the most challenging ones was increasing leaf size, so this is often difficult to determine visually, but this is whether or not there are current or expanding new leaves on the page. And of course, colored leaves, flowers or flower buds present on the page, fruit or seeds, and ripe fruit. So we were able to pair this assessment of the phenological activity depicted on the press specimen with the label data that often has information about date of collection and location of collection. And because we use the same protocol, we're able to easily pair that with the ongoing data collection occurring from the Nature's Notebook and the National Phenology Network. So when we compare those two, so what we did here was we limited the herbarium specimens to just the ones since the 1990s, because we didn't want to be skewed by changes over time in the occurrence of these phenophases, and compared the proportion of herbarium specimens showing each phenophase with the proportion of National Phenology Network's submitted observations that had those same occurring phenophases. And while I've highlighted some key inconsistencies in the colored leaves, the flower buds, and the fruits, we actually found that there was extremely good parity between the different observational strategies. And this gives us some strength, some oomph behind our analyses to say this is actually something useful that can be done to extend the citizen science record. So Nature's Notebook doesn't go back past the 90s, but this allows us to have data that goes back even further. So when we put these together, we have a data set that decadally can go back to the late 1800s, showing trends in the expression of these phenophases per month. So what this graph is each month is graphed with one color as a weighted regression with the number of samples as the weight, and each year is its own data point. So in the, let's see, so up here, for example, each of these purple dots represents the percentage of specimens early on, and then later on the percentage of citizen science observations that were showing colored leaves in each decade. As you can see, it increases over time. So there's an increase in the proportion of these specimens and observations that are showing colored leaves in the 10th month over time, which is extremely interesting. If you want to know more about the specific things we found using this data set, I have a poster that you can watch at your convenience. But in short, this is just a demonstration of the ways that pairing citizen science data products with these herbarium-derived products allows you to look for long-term trends. Another example of what this might look like is the Opal Air Quality Survey. And so this project was put in practice in the UK primarily, where citizen scientists went out and looked at the lichen and the leaves in their community to help determine how well it matched to their current air quality conditions. One of the major metrics they used was the presence of leaf tar spots, which is a type of fungus, on sycamore leaves. And so down here are some images of what, down on the lower left are images of what that might look like in life. So these leaves with these dark black spots. But these are actually preserved in herbarium specimens as well. So over on the right, you see this is an herbarium specimen that has the presence of these leaf spots. And the same way that the protocols are used for citizen scientists in the field to collect data, counting the number of spots per leaf, counting the number of leaves with spots per single occurrence, is a second example of how a citizen science protocol could be used to collect actionable information and extend the usability of citizen science products while extending the use of herbarium products. So these are just two potential examples. There are many more. The number of projects and citizen science opportunities as well as protocols are growing. So as these programs continue to increase in scale and scope and size, the number of available protocols that can be reused as protocols to do visual assessment of herbarium and museum specimens are continuing to grow as well. The other side of this is when you have protocols that are well known to citizen scientists in the wild, quote unquote, these protocols can also be used for those same citizen scientists or similar citizen scientists to be scoring the herbarium records for you. So one of the things we're exploring now are using programs like Notes from Nature to score herbarium specimens using these same protocols and targeting the same communities to allow for similar observer statuses across the board. So in short, harnessing the citizen science protocols and communities for usage on digital herbarium and museum specimens allows us to tap into an existing community and existing knowledge base while increasing the opportunities for integrating data across different time periods and localities, and considering the ways that we can explore and understand better the environmental trends present in these museum specimens beyond just a record of occurrence. So overall, I believe that applying these protocols can allow us to better understand the world around us while increasing participation from citizen scientists and researchers in the data collection and analysis and understanding process. So of course I have many funders and collaborators to thank and acknowledge, and I really appreciate especially the support of George Mason and my collaborators there. And at this point, I'm happy to take any questions.